school. My mentor was Doctor, and I was in her. And the title of my presentation is 2F51 High Resolution Structure of Recombinant Trichomonas Vaginalis Thioredoxin. It's not as complicated as it seems. Trichomonas vaginalis is another word for trich. It's a sexually transmitted disease, and it's caused by the parasite called Trichomonas vaginalis. It's an anaerobic organism, and it's unable to survive in the mouth or the rectum. Therefore, it can be transmitted through penis to vaginal intercourse. And this is cured by, it's curable. It's one of the most common STDs that's curable by metronidazole and tenofovir. It's given by, it's a single dose, and it takes time for it to cure. And thioredoxin is a oxy-reduced enzyme. It catalyzes a wide variety of redox reactions in the cell. Redox reactions are a type of chemical reaction in the cell where one reductant is oxidized and the other one is reduced. The structure of thioredoxin from trich is at a resolution of 1.9 angstroms. The structure has a 5 beta sheet structure with 2 helices on each side. And it has an active site, and that's the protein tyrosine, cytosine, glycine, protein, cytosine, and a motif of 34 to 38. This is just basically talking about where the side chains of the of thioredoxin are. This is the structure, and right here is the active site of the protein, and these yellow arrows, they are the beta strands that make up the beta sheets, and these really looking thingies, they're the helices, and there are 4 of them. This is a primal image of the structure. This is the comparison to human thyrodoxin. It compared to human thyrodoxin, CBTRX, that's another name for the thing. It has addition. It's longer, and it has an addition of 3 amino acids right here. And right here you see the sequence, and the red background is conserved strictly of 2 proteins, and the white background show that it's homologous. Alright, this is the methods for the experiment from the research paper. Basically, the people they found the TRX gene. It was expressed in a sequence, and then the tag database was cloned and inserted into a vector to create the plasmid. The plasmid was then put into an E. coli strain so it could be for protein production, and then the single colonies of BL21, which is the bacteria that has the plasmid, which is the BPV1, were grown in the Luna Bertini medium, and then they were expressed, and then after that they were purified for crystallization. And this is the materials: the vector, the plasmid, the E. coli strain, the median, the glycoside, the phosphate, and the matrix acidic matrix. This is how I basically got all my information for all my research. I went to PDB, which is protein data bank, and I typed in my code 2F51, and I got the research paper, and I did a blast on this protein, went to the NCBI homepage, typed in blast, and copied the FASTA sequence, got the sequence and put it and blasted it against non-redundant proteins. This is BLAST. It's a basic logo alignment search tool. It's used to show the alignments in the protein, and so you see which structures are similar, like in monkeys or like chickens and stuff. Alright. I then use the Ramacharyan plot. It just basically shows the concentrations of the beta sheets and the helices in the protein. It just tells you where to make it simple, it just tells you where the beta sheets and the helices are in the protein structure. And this is it, right? Alright, right here you can see is the beta sheet. Right here you can see the helices. And it's not that complicated. There's more. Basically, the whole Ramachandran plot shows that 97.3 of all the residues were favored in 98% of the regions, and 100% of all the residues were allowed in greater than 98.8 of the regions. Compared to other thyrodoxins, it's found that TBTRX is most similar to human homolog, and it's basically, it has 14 side chains, and as you can see, it's self-explanatory. Alright. In the future, I hope that the rest of the conformations are modeled in order to help us further understand the structure of this protein. By doing so, we are able to find even more effective ways to cure this STD, and to maybe understand how we go about studying other STDs. Carl in his class.